Greetings. This is Biology Paper 2 Theory, and this is the paper for the GCE's 2025. I want to make it simple so that even as you get to prepare for your exams, you are going to learn a lot from this particular question paper that I'm going to solve. So this should give you an idea on how you are going to answer your paper. Quickly, let us jump into the questions so that you get to see them and even as we get to answer them together. So quickly, we jump to question number 1. So this is section A that I'm going to answer, and the question is saying, short answer questions, 44 marks. Question number 1 here. So it's saying answer all the questions in the spaces provided in the question paper. So question number 1, figure 1 shows a parasite cell observed under a light microscope. So you know that the function of a microscope is to see those tiny objects that cannot be viewed when you use your naked eyes like that. So this is a parasite cell. There are parts labeled there, so to make things so short, since we are revising through, we are able to know these parts. The good part is that the first question, they're even telling us to label those parts. So identify the parts labeled A, B, and C in figure 1. So what is the name of this particular part? This is the simplest, and I don't expect anyone to fail. So the first one, that is a cell. So A, that is the cell. Then the B, have you seen this? That is what we get to call the vacuum. So that is the vacuum. So that is it. Then we go to C. C, have you seen where C is pointing? C is pointing at this thing that is there. That thing that is there. That is the what? This one that is here. This is the what? The chloroplast. So I'll say that is the what? That is the chloroplast. So you are done just like that. They give you these 3 marks. Then this question is telling us, question B, explain the functions of the parts labeled, functions of the part labeled A. What is the function of the part labeled A? That is the what? The cell. What is the function of the cell? The function of the cell, you need to understand that it is us to protect. So you are going to say that protect the what? Protect the inner part. So I'll say protect the inner part of the what? Inner part of the cell. It protects the inner part of the cell from damage. So from what? From damage. This is it. So we are done with that. We now go to this next question. Give 2 differences between the cell in figure 1 and the one found in the human trachea. So this is a particular question that you are given here. As you can see, this one, the cell in figure 1, this is figure 1, this is what? This is a plant cell. And they are telling us the one that is found in an animal cell, human animal cell. So they are just telling you to give some differences between animal cell and a plant cell. So what are those particular functions that you get to observe? You will discover that you say that figure 1, so you say figure 1, you know that is a plant cell. Figure 1 has what? As chloroplast. So figure 1 has chloroplast. Then you say, wow, human trachea, like this, doesn't have a what? Trachea cell doesn't have. So does not have. We are just giving some differences there. Then the other difference is where we get to talk of the cell. Have you seen? This one gets to have a cell. But if you check an animal cell, it starts with the cell membrane. So meaning that you are going to give a difference by saying that figure 1, so I'll say figure 1, figure 1 has what? Has a plant cell. Cell. It has a cell hole. Wow. Cell. So cell found where? The cell found in the what? In the, so have you seen I'm running out of space here? So I'll just say in the what? In the tracheal has no cell. So it doesn't have any cell. Has no cell. So this is a difference. You are done. So this is what you are supposed to put there. So the difference is, if this plant cell gets to have, has chloroplast, while human trachea cell. So while human trachea cell does not have. Figure 1 has a cell, while cell found in the human trachea has no cell. You are done with that. We now go to D. I guess you are able to see part D. State the names of any two organelles that would be seen if the cell in figure 1 was observed under an electron microscope. So they are telling you now, if we get to view this particular figure under a microscope, which is an electron microscope, you discover that we are going to see some detailed parts that we are not able to see. Those of you that are not able to view this clearly, you are not able to see these weights, you can make your screen so big where you are watching it in landscape, you are able to see everything that is here, so that you don't trouble your eyes if these weights are too small. So what are those parts? We've got ribosomes. So you get to see them under the what? Under the microscope, detailed structure. So we get to see these. So say ribosomes get to be seen. Then we also get to have the mitochondria. So I'll say mitochondria. So mitochondria, something like this. So this is it. So I guess we've seen these are some of the parts that you are going to see. We've got such parts like Golgi bodies, those particular parts. So let us make sure that we are able to study even the detailed structure of a plant cell and the animal cell. Questions do come in an exam, not forgetting cell specialization. So all those things, you need to know them. You need to know different types of specialized cells and their adaptation. So I proceed. I guess you are enjoying this even as we are moving together. So we go to question number 2. This is question number 2, a good question. Figure 2 shows stages involved when testing for the presence of a food nutrient W in crushed onion. So this is what is given. Use warm water, crushed onion, put there. They put reagent X there. In their vision, they are putting reagent X. Then droppers like that. Stage 3, they apply heat. You discover that here they get to have a lead precipitate. So crystals that are made of color lead get to be formed there. These are red precipitates. And then there is water there. All these things, you get to study all the stages. To answer this particular question, you need to know a topic that is called a food test. There is such a topic in biology, even as we are starting. So you need to know it. You need to know how to test for starch. You need to know how to test for reducing sugars. You need all those food tests. You need to know them. So even as you look at this particular part here, they are telling us to say lead precipitate is formed. So we can even now conclude that here this was reducing what? Sugars. Now the question here, identify reagent X. What is this reagent X? Reagent X, so when we are testing for reducing sugars, a solution that we get to use is called a Benedict solution. So we get to use the what? We get to use the Benedict solution. So that is reagent X, the one that is there. So you just say Benedict what? A Benedict a solution. Then they are telling you to say, explain why the onion was crushed. So give a reason why we get to crush the onion and shaken with warm water. So they're asking us to, first of all, we start by knowing the reason why we get to crush onion. So you discover that the onion is crushed so that we get to disrupt the cell structure. So onion is what? You are going to say onion is crushed. So onion is what? Onion is crushed. So onion is crushed to do what? To disrupt. So I'll say to disrupt the what? The cell structure. So the structure of the cell. Structure of the cell. So structure of the what? Structure of the cell. This is it. Then comma, then this part now shaken with warm water. So it is shaken with warm water. So I'm going to say that shaken with what? Shaken with. So shaken with warm water. So shaken with warm water. Warm water to do what? To extract. So that we can do what? So that we can extract the what? The sugar from onion. So we can extract the what? We can extract the sugar from what? From onion. Because this is testing for sugars. So this is it. Onion is crushed to do what? Disrupt the structure of the cell. Shaken with warm water to extract the sugar from what? From onion. Like this, you are done. And even as I'm answering like this, you need to know that biology is so complex, a lot of things get to be involved. So a person can come in with another view, which is also correct. Then we now go to C1. C1, they are telling us to say that from the results in stage 4, where is stage 4? Stage 4 is there. So stage 4 is there. They are asking us to say, which nutrient in the onion was being tested for? So which nutrient in the onion was being tested for? So I've told you reducing what? This is reducing sugar. So we just say what? Glucose. So just put glucose there. Then we go to 2. They are telling us to say, name the class to which the nutrient identified in C1. So the nutrient which is glucose belongs to what we are get to go. So which group of the nutrients? So there you know that we are talking about what? The carbohydrates. So we are talking about the carbohydrates. So to be specific now, these are carbohydrates that we are talking about. These are the what? Monosaccharides. So monosaccharides like this. So this is it. Kindly confirm the spelling with me. So kindly confirm this particular spelling with me. Then we go to this question. Name one other class of nutrients found in onion other than the one identified. This one that is here. So the other one, we can talk of what? Vitamins. So we can talk of vitamins. Aside carbohydrates, we can talk of what? Vitamins. So this is it. Then we proceed and go to the last question, and question number 2. What are we taught there? The question is just telling us to say, explain the importance of food nutrient W in the human body. Food nutrient W in the what? In the human what? In the human body. So now they want us to do what? To explain the importance of food nutrient. W in the what? In the human body. So you are supposed now to understand that the importance of food and nutrient is that. So first of all, you know what we are testing here. The question there was telling us to say that involving the figure 2 shows, so the diagram here, it was saying that, have you seen all these things that are here? The question was saying shows the stages involved when testing for the presence of food nutrient W. So the presence of food nutrient what? W. Now here, since we've identified what the nutrient is and everything, so what is this for? You discover that the importance is that it is important in so many ways, because this is you say it is important because this is the source of what? Source of energy. We talk of source of energy, so this is a source of energy for what? For growth, where we get to look at growth. We can also look at other things like breathing. So breathing, we look at breathing, and it is also necessary. You need to understand that it is also what? It is also necessary for what we get to call some proper what? Proper secular, so proper secular function. So this is it. This is what you are supposed to know. This is what you need to understand. So we are done with the question number 2. And I believe you are able to see how I'm answering this particular question. Let us now dive to question number 3. I guess you are able to see how I'm answering out these particular questions, and if you are to meet such questions in an exam. So I'm just giving you some hints. So all these topics that we've seen come. Here, this question is saying figure 3 represents 3 different models of gaseous what? Gaseous exchange in different animals. So here they are teaching us, or they are testing us on what? Gaseous exchange. If we are able to differentiate them from different types of animals, how this thing called gaseous exchange, you need to know how it occurs in people, how it occurs in fish. You need to know how it is in insects. All those are things you are supposed to know them. So this is to tell you that immediately after attending to this lesson, you need to go through those things. Now here, which model represents gaseous exchange in human beings? So in human beings, here, just know that whenever we are talking about blood capillaries, just know that that is what? Human beings. So you say that is model what? Model 1. Then insects. So when we are talking about insects, it is connected to what? It is connected to tissues. So it is connected to what? To tissues. So that is model 3. So that is what? Model what? Model 3. Same way it is written, that is model what? Model 3. Then we proceed to the next one. Give reasons for your answer in A by referring only to the what? Models in figure 3. Just these ones that are here. So why have we said that this one belongs to the human beings? It's because of what? You say because of blood capillaries. So I'll say blood what? Because of, so I'm going to say because of what? Because of blood capillaries there. So I'll say because of blood what? Blood capillary. So I'll say because of blood capillary and the direction of gases. Have you seen? Said okay, and direction of gases. So and the direction here they just want one answer, so you can pick direction of what? Gases, like that. And then we go to the next one. Insects. How have we identified that this is insects? Because of what? I've told you, because of what? Because of tissue cells. So because of what? Tissue cells. This is the reason why I'm able to identify that this is this This is that. We go to C. C1, we move to a C1. C1, which model in figure 3 uses water as a medium of transport for oxygen to the respiratory surface here? So uses water. So since we've said that human being, we've said this is from human being, this is from insect. This one that is there, that is what? Fish. So we just say here, they're telling us model. So I just put model 2. I know that that is fish. So model what? Model 2. So this is it, model 2. Then they're telling me, name one living organism which uses the model that you've identified. It is what? It is fish. So I just say a fish there. I put fish. Then I go to this question that is here. This question saying, suggest structures that head what? So they are telling me to say, suggest structures that help movement of air into and out of what? In this part, which is called 1. Have you seen 1? I start with the model 1. So that helps head movement of air in and out of model 1. Human beings there. So they want us to suggest. So I just suggest how many here? I suggest 2. So I can talk of rib cage. So I can also talk of rib cage there. I can also talk of the diaphragm. So I can talk of the what? A diaphragm. So kindly confirm this particular spelling for the diaphragm. This is it. So rib cage, we also need to look at the diaphragm. Human beings, I guess you are able to understand that. Let us now go to the next one. So model 3 there, the question model 3. Model 3, we know that is what? Model 3, we've identified that is insect. So in insects, what are some of the things that you need to get there? Here, only one answer there. So just put one answer there. So you can talk of the trachea. If you remember where we are coming from, you understand all these things. So you need to make sure that you are able to identify them. Let us now go to this question that is here. This is a question saying, figure 4 is a bar chart showing the volumes of urine collected from a person before and after drinking. This is 1.5 cubic decimeters of water. The person's urine was collected immediately before the water was drunk as indicated at time 0 and then at 30 minutes intervals for 4 hours. So this is a bar chart that we are given. Have you seen? This one is a what? This one is a bar chart. So we are given this particular bar chart. Then we are given the questions here. We've got question number 1, which is A1. Explain why there was a high volume of urine after what? After 60 minutes, after 1 hour. So explain. We are told now to explain. So this is the answer. The network, the phone had to misbehave. So this, as I was writing this, it was not being recorded. So this is the answer here, where you are told to say, explain why there was a high volume of urine after 60 minutes. Have you seen after what? 60 minutes. Here it went, like that. The reason is that because of the fluid in the bloodstream, so the kidney had to absorb less water back into the bloodstream, which resulted in more water being extracted. So this is it. So this is how we are supposed to answer it. We proceed now to this one that is here. Now this question is saying, state the point at which the volume of urine stabilized, where the urine now had to stabilize. Have you seen? It was here. Have you seen here at 210? It started at here. Have you seen? This is how it started. So if you check like this, here it had to stabilize. So it is what? 210. So the answer there, you just say 210. So you say 210 minutes. This is it. So 210 minutes. Say 210 minutes there. And then name the organ from which the urine was being excreted. So the organ there, we can urine. We know that definitely it is the what? It is the kidney. Then the next question: what process is being demonstrated? The process that is being demonstrated here is what we get to call osmoregulation. What is osmoregulation? When we are talking about osmoregulation, this is the regulation of water molecules in the body. So we are talking about water here. Then we proceed to this question that is here. The question is telling us to say, explain two other processes by which the water drunk would be lost from the body. How do we get to lose water? So you discover that water gets to be lost via a lot of things. We can talk of there is what is known as a skin evaporation. So we can talk of skin evaporation. We can also consider even sweating. So even sweating can be a factor here. This question that is here telling us to say which conditions would increase the loss of water from the body. So the conditions, high temperature. So when you're having high temperature, we can increase the what? The loss of water from the body. We can also talk of low humidity. So these are the answers that you need to get and understand. So we've answered this particular part. Finally, now we are on the last question. We are now on the last question. So I believe and guess we are all going to understand the last part. We know that always question number 5 is under genetics. How do we answer this question? How do we go about this particular question? A man who had normal vision married a woman also with normal vision. They had a color blind son. Color blindness is sex linked. Using arrows, so we are told to say using arrows. So here we've got this one, have you seen? We've got this one B, so meaning dominant, this one recessive. So when you are having this meaning normal, then this one sick. But whenever we are talking about a sex linked, you need to understand that you just get to have X for a man. For example, when we are talking about a man, you understand that a man gets to have X and Y. So this is to tell you that a man cannot be a carrier because a man gets to have XY. So meaning that a man, if the person is like this, have you seen? Meaning that is a normal man. But if it is like this, meaning that that is a person that has got this color blindness. So, but a woman, when we are talking about a woman, they get to have XX. So if the person is like this, have you seen? It means that the person is what? The person is just okay. But if a person is like this, for example, like this, then XB. The person is normal, but that particular woman is a carrier, and this same blindness can be seen in the offspring. So this is what you need to understand. So now, going to this particular question, predict the genotypes of the man and his wife. Genotype, genetic makeup of an organism. So a man, this is a normal male. So what we are saying that it is just X, capital B and Y. But this, for them to have a son who had for them to have a color blind son, meaning the woman was a carrier. So meaning that is X, capital B, X, small b. So this is it. this person is normal, but the person is a carrier, and these characteristics can be seen in the offspring. So if the person is normal but is a carrier, this one, if you are a man, you've got the color blindness in you, or you are just normal. There's nothing like you are a carrier. No. Now we go to this question. They're telling us to say, draw a genetic diagram to show how this was possible. They want us to put a genetic diagram to show how this thing was possible. You understand that the first thing we get to start with what? We start with the parental phenotype. What do we mean by phenotype? Phenotype, this is the outward appearance of an organism. So this is, we say a normal what? A normal man. So a normal man crossed with what? Crossed with a normal woman. So even a normal what? Normal woman. The woman was just okay. So I say normal woman there. Then I say parental. This is a genetic diagram. Genetic genotype now. Parental genotype. So parental genotype now, where we know that the man, that is X, B, Y there. Then woman, carrier, like this, then X, small, B, like that. So this is it. So you can put X there. This X is very important because you are able to show that we had to, like there was something that happened between them, which is sexual intercourse, of course. So now, here, the next is where we split these to form gametes. So meaning we split them so that we get to form what? Gametes. So gametes. We get to form gametes. So this is X, B there. This is Y there. This is X, capital B, X, small b. So we are crossing these things. So what you need to get now, fertilization now gets to occur, where this one, you bring it down here like this, then it goes there. Some people don't know how to draw genetic diagrams. Discover that they just put some things that get to confuse the markers. X, B, X, B. So here, what are we forming here? We are forming F1. So you say F1 offspring. So F1 offspring here. And this is what? Genotype. So F1 offspring genotype. This is genotype. So this is genotype. So meaning that down here, we are going to form F1 offspring phenotype. This one first comes here. Here. So here like this. It goes there. There. So that is X capital B, X small b. Like this. Then here, this one comes here. Like this. Then goes there. Like that. So meaning that this is, we start with the X, capital B, Y, like this. Then this one goes at the end there, then it goes straight like this. Have you seen how my genetic diagram is? So X, small b, and Y. So this is how it is supposed to be. Then here I say this is what? F1 offspring. So F1 offspring. So F1 offspring like this. Then this is what? Phenotype. I now put a phenotype there. So this one, this is a girl. So this is a normal what? A normal girl. So I say this is a normal girl. This one that is here, this is a normal girl, but a carrier. Have you seen? This is a normal girl, but a carrier. This one, this is a what? This is a normal son. So this is a normal what? Normal son. Because of XY. Only girls get to have XX. So this one, this is also, this one is now a color blind son. So you say a color blind what? Blind son. So just by looking at this genetic diagram, you are able to tell that it is possible to have a son with the color blindness. Have you seen here? It is here, color blindness. So here, it is very possible. So just this simple diagram, they give you free 5 marks. Can we fail to do this in an exam? No, you can't fail. So let's make sure that we are able to practice through genetic diagram. Let me just say genetics. Make sure that you study it, understand it, so that if questions are to come in an exam, you will be able to answer them. Let us now dive into the last questions. So let us now go to the last question. So it is here, the last question now. This is the last question. One of the causes of color blindness is mutation. So this is a cause of color blindness. They are telling us to say now, define a mutation. What is mutation? Mutation, you just say that this is the word spontaneous or the sudden change. So you say this is the sudden change in the what? In the structure. So in the structure, I'll say in the structure of the what? Structure, I'll say in the structure of the DNA. Structure of the what? DNA, or number of chromosomes. Or number of what? Chromosomes. So number of what? Chromosomes. This is it. So this is what you need to get there. Then they're telling you to say that explain one cause of mutation. So what is this cause of mutation? So one cause of mutation, we can talk of radiation. So you discover that radiation, the rays that can also cause, so when we talk of radiation, such as what? Such as we can talk of X-rays, gamma rays. So we can talk of what? Gamma rays, like that. So etc. So this is it. You just need to explain. Radiation. Radiation, what we mean is we talk of X-rays, gamma rays, like that. So these can cause what? Mutation. We've come to the end of this particular session. I guess you've enjoyed, and make sure you subscribe to this particular channel page, like, comment, so that each time I upload a new content, you are going to see it, and you are also going to help us have a larger audience. So support me, and make sure that if you want tuitions, you text me on WhatsApp, where I get to do online lessons at an affordable fee. So if you just want this one, which is biology, it is only 100 kwacha monthly. So are you a GCE? Are you writing your exams? Let me help you. Do you know anyone that is struggling with mathematics, science, biology, RE 2046, English language? I'm here to help you people get the distinctions that you need. So don't forget to get my number. You pay, I add to the group. This is my number, where I get to teach not just these papers that you are able to see. I upload notes. You understand them, then we revise through together these exam questions. So this is the end. And please let me know where you are watching me from in the comments section. So I'm able to see the comments. Just tell me the location where you are watching me from. Thank you so much.